All right, welcome everyone. Thanks for coming to today's Dream CPAR seminar. We're really excited to welcome Professor Victoria Webster-Wood, who is an assistant professor in mechanical engineering at CMU. And she runs possibly the coolest sounding lab I have ever heard of, the Biohybrid and Organic Robotics group, the Borg Lab, and is going to be talking to us today about these, bridging these different areas of bio-inspired and actually biohybrid robotics. And her work has been featured in a number of places, including Popular Science or Popular Mechanics, excuse me, and NPR. And she just received an NSF Career Award, which is super exciting. And I'm really excited to hear about living machines today. So take it away.
Thank you, Laura, and thank you everybody so much for having me with you today to share our work. Like Laura said, I'm a third-year assistant professor at Carnegie Mellon University, and today I'm going to be sharing with you all the work that my lab is doing in creating living machines. And our goal is really to bridge between more traditional bio-inspired robotics and move into this emerging area of biohybrid robotics. And so as we get started with this, there we go. One thing that has really motivated me in my career in robotics is just these amazing feats that animals are capable of. They demonstrate incredible agility and adaptability in complex, dynamically changing environments. That this mountain goat video is one of my favorite videos, where these mountain goats are climbing on these essentially sheer cliff faces, caring for their young, looking for food, avoiding predators. And animals are also capable of tremendous problem solving. Crows and ravens have been shown to understand how to use tools to achieve their goals. And even in our oceans, we see these tremendous feats of agility and coordination in the movement of animals in these dynamically changing complex environments. And this is really what we want to be able to achieve with our robots. And there's kind of a continued need for mobile robots to translate out into real world environments, and in particular hazardous or sensitive environments where maybe we don't want to have human presences because it's not safe for the human or it's not safe for the environment. And in particular, in looking at how bio-inspired robotics can help us in these spaces, I'm really motivated by the need for low-cost robots for things like search and rescue, where you have a very dangerous environment and you're very likely to lose your robots in the process of deploying them in these type of rubbles, which may then further impact the environment, as well as environmental monitoring, where we have these very sensitive ecosystems that we'd like to be able to monitor and even provide maintenance for. But we need robots that we can deploy in these environments that aren't going to further damage the ecosystem. And then there's also kind of emerging spaces in medicine where there's a tremendous need for small-scale robots that can safely navigate through the vasculature of living organisms to provide targeted drug delivery or maintenance and monitoring of these vascular networks. And in each of these environments, we really need robots that can safely interact with living organisms. And so my lab is driven by this question of how do we capture these amazing abilities that we see in animals in safe, robust, autonomous robots? And when we look at this question, we see that we have some key system needs to achieve this goal. We really want our robots to have some level of compliance so that when they come into contact with living organisms, they're not going to do damage. We want robots that are energy efficient so that we don't need to have a tether or heavy onboard power supplies. These robots, of course, need to be autonomous, and with that comes a certain level of behavioral flexibility, or the ability to adapt their behaviors in response to changes in their environment. And when we're thinking about deploying robots in the real world, I really encourage everyone to think about how can we deploy robots in such a way that they'll have minimal negative impact on the environment we're deploying them into. Now, unlike traditional robots, these animals that we're taking inspiration from, these biological systems, are composed of dynamic, responsive materials. For example, tissue growth is guided by biodegradable proteins that can actually be remodeled by the cells in a given tissue in order to adjust the tissue properties in response to the loading conditions of that tissue. When we think about actuation, in biological systems, the actuators are muscles, which are composed of these functional fibers in a very highly organized architecture that is also capable of adapting to the mechanical or electrical stimulation and loading that it experiences. So your muscles can get stronger as you exercise them. What's interesting about muscle, when we think about robots, is that it's really a kind of good enough actuator in terms of many of the desirable robotic actuator metrics. And while it may not exceed any individual specialized actuator in one area, by providing kind of these good enough capabilities across the board, it's really intriguing as a source of inspiration, and as you'll see in the course of this talk, as a possible material itself for robotic actuation. At the control level, biological systems are controlled by these complex systems of neurons and synapses that have complex structures and functional regions that are all highly interconnected. And again, these connections and connection strengths can adapt in response to experiences and enable learning. And so when we come back to these system needs for our safe, robust, autonomous robots, we see that there are already many solutions in nature. Of course, many animals, such as this jellyfish or this octopus, are composed of completely soft materials and actuators. Muscle as an actuator is relatively high power to weight ratio, and many animal systems are capable of lifting weights even above their own body weight. Animals are capable of tremendous feats of asynchronous parallel control. So we're able to balance all of these competing needs and process all of these different goals in real time. And we can do this in a way that we can learn and adapt. And in nature, even something as simple as this single-celled slime mold demonstrates learning and plasticity. And of course, with all of these biological systems, because they are made out of these living materials, they're all naturally biodegradable, which minimizes the environmental impact of these animals in their environments. And so this is what has really motivated the work that my lab does, where we're seeking to kind of start from a place of bioinspiration and start from bioinspired robotics and move towards the ability to use living and biodegradable materials directly as engineering materials in our biohybrid robotics. For us, this begins by considering sources of renewable biocompatible structures, such as using those extracellular matrix proteins directly as scaffolding materials for our soft robotic systems, such as the use of electrocompacted collagen, which we'll be talking about today. And we can use this to create kind of these robust scaffold materials. With these materials in mind, we can then look at developing living adaptable robotic actuators using muscle directly as the material for the actuator. And in my lab, we take two approaches to developing living actuators. One where we kind of use tissue engineering techniques to grow the actuator tissue, and another where we're able to harvest actuator materials from farmable organisms. And our work in developing these muscle bioactuators, as we'll discuss later in the talk, has really motivated the need to consider not only the actuator, but also the motor controller, and to think about developing living motor controllers composed of neurons. We're very interested in this both in terms of using explanted neuromuscular tissue circuits, where we can maintain functional connections to the nervous system, as well as growing our own neural circuits with living neurons in the dish. And this is all towards our long-term goal of creating renewable, biodegradable, autonomous biohybrid robots. And as we've built this bridge from bio-inspired robotics to biohybrid robots, and we've studied these tissues and model animals along the way, what we found is that we actually kind of create this interesting feedback loop back to bio-inspired robotics. For example, our work in kind of studying these tissues for living bioactuators has allowed us to start developing simulated and physical neuromechanical models using bio-inspired robotics techniques to create and improve upon kind of these neuromechanical models of animal models for use in neuroscience. And our work in understanding how we can develop these living motor controllers has led us to develop techniques in brain-inspired computing, both for real-time prediction of neural activity in these controllers, but also as a source of bioinspiration for approaches in artificial neural networks. And so in today's talk, I'm going to be presenting our work in each of these key areas, starting with our efforts in developing renewable biocompatible structures composed of electrocompacted collagen. I'll also share 2 approaches that we take in the creation of living adaptable robotic actuators, and discuss some of our recent work in brain-inspired computing, both in the development of these real-time models for neural circuit activity, and in an approach for forming the connections for artificial neural networks that we've dubbed growth-initiated neural networks. And I'll wrap up by discussing some of the future directions that we see in biohybrid and bioinspired robotics. Of course, before I dive into the actual research, I would be remiss if I did not give a shout out to the people who are actually doing this research these days. We've got a fantastic team of PhD students, master students, and many undergraduate students who've come through the lab, even over the course of the 3 years that I've been here. And of course, this work would not be possible without their efforts. So let's start with our work in renewable biocompatible structures. And much of this work for us is centered around this electrocompacted and aligned collagen-based biomaterial. With this material, what we do is we start with a molecular solution of collagen molecules and place this between 2 electrodes. You can then apply a voltage across the electrodes, and the collagen molecules will become charged. This results in forces on the molecules, which cause them to migrate and compact along the isoelectric point. And in doing so, they actually form really robust structures. If you do this with wire electrodes on either side, you're able to form an electrocompacted and aligned collagen thread. And if you do this with plate electrodes, you can form sheets of collagen material. These can then be further cross-linked to increase their mechanical strength, even to the point where they can be used as sutures. And so what my PhD student Wenhun Sun was interested in is how can we really kind of develop a system for using this material as an engineered material in our biohybrid robots. And in order to do that, we really need to understand how the fabrication properties involved in this isoelectric focusing impact their mechanical properties in order to be able to get materials with the mechanical and geometric properties we need for our robots. So what Wen did is he looked at how the impact of compaction time, compaction voltage, and collagen concentration impacted the mechanical and geometric properties of the resulting threads, with his goal being to see if we could really tune these properties and predict the properties a priori. And what he found is that there is really some emerging relationships between these fabrication parameters and the thread properties. For example, if you apply this isoelectric focusing effect for a longer amount of time, it increases the failure strain of the threads. Similarly, longer isoelectric focusing durations also result in thinner cross-sectional area to the threads. And so you can really tune both the mechanical and geometric properties. Conversely, if you increase the voltage of the isoelectric focusing compaction, the cross-sectional area of the threads also increases. And overall, he found that really he could tune the mechanical properties of these threads over a wide range of tensile moduli and tensile strength. And actually, he's going to be presenting at the Society for Biomaterials meeting later this month on a deep neural network tool that he's now developed in order to tell you what fabrication parameters to use in order to achieve specific material properties that you want in these sheets and threads. And why this is so interesting to us for building our biohybrid robots is that it turns out the alignment of the collagen in these structures can be used to guide the self-assembly of bioactuators. So if we take this collagen and compact it using plate electrodes, we can compact sheets of the material. When you first compact the sheets, you get this kind of randomly organized collagen structure. So here, this is a polarized light microscopy image where the blue indicates alignment kind of along this 45 degree. And so you can see there's not a lot of blue here, so the sheet's not well aligned. And if you grow muscle cells on that material, the muscle cells are kind of randomly aligned, right? But if you remember back to the earlier slide, muscle is kind of a very highly organized tissue with these aligned fibers that contribute to the contraction force. So we really want to be able to reproduce that in our actuators. Turns out that if you stretch these sheets, and you can kind of mechanically induce alignment of these collagen molecules. So here we're seeing again this polarized light microscopy image, and this blue color shows us we have much more alignment along that 45 degree. And we can tune the degree of alignment by how much we stretch the material. Now if we grow cells on it, the cells become highly aligned. And therefore, as the muscle tissue is forming, when it contracts, all that contraction force will be aligned together. You get a much more effective bioactuator. And this really led to our first efforts in creating these living adaptable robotic actuators and the development of cell-powered organic robots. So building on this work in electrocompacted collagen materials, we were able to fabricate scaffolds for these robots kind of en masse using this electrocompaction by fabricating molds with the desired body shape for the robot and applying the compaction technique previously described to the collagen, which results in the structures of the robots being compacted onto the cathode during the electrocompaction. We can then peel these robot bodies off of the electrode, which has the effect of both removing the robot body from the electrode, but also kind of stretching the legs, which induces that alignment of the collagen. We then seed muscle cells onto the bodies of the robots. And we've done this with both cardiomyocytes isolated from chick embryo hearts, as well as skeletal muscle from these chick embryos. Then the passive stresses of the cells actually result in the robot taking on a 3D conformation, which we can then flip over to get our robotic device. And just like we saw with the basic sheets, that stretching process really does induce alignment of the collagen along the legs of the robots, which we verified with scanning electron microscopy. And this then aligns the cells along the legs of those robots. Now, so we've got this essentially bilayer cantilever with muscle cells on one side and the collagen material on the other. And when the muscle cells compact, the result is that the cantilever bends. And we can paste this externally with an electric field, and in this way, get the robots to move. So what we're seeing here, this shadowy cloud, if you will, is the actual robot. We're pasting it with an external electrical field, and you see slowly over time in this time-lapse video, it is creeping forward. Now, of course, I should highlight, this is at 10x speed. These robots are very, very, very slow. And we tested this with both cardiomyocytes and skeletal muscle. Overall, both types of robots using this approach were relatively slow, but the skeletal muscle did kind of improve the device performance over the cardiomyocytes. And so this shows us that we really can kind of self-assemble these bioactuators in order to generate locomotion in these robotic devices. But it also led us to kind of an engineering challenge, right? How do we improve the device robustness for any of these applications that I was talking about beyond the lab, right? These robots are extremely slow, and they also require really specific environmental conditions to function. They require a specific temperature, pH, salinity in the liquid. You have to feed the robots because they're living materials. And we were really interested in how we can improve that robustness. And this really led us to our second approach in creating these adaptable living adaptable robotic actuators. And this was to look for a more robust source of material for the robots, which led us to Aplysia californica. Aplysia californica, shown here, is a robust marine sea slug. It lives in the intertidal region off the southern coast of California, where it lives in these tide pools. And so it can experience these really large temperature and osmotic swings as the tide goes in and out. For example, it lives at about 16 degrees Celsius, but the tissue functions at and even slightly above 25 degrees Celsius, which is really quite amazing. If you or I had a fever of 9 degrees Celsius, we'd be in a lot of trouble. So this is much more robust tissue. It has very large, robust cells, which have also made it a model system for neurobiology research. And so there's a lot known about how the neuromuscular system is organized and how it functions. And this work really started while I was at Case Western Reserve University doing my PhD. And so I really want to acknowledge the great team of students that I was able to mentor there as well who've contributed to this work. In order to use Aplysia californica as a farmable source of these more robust bioactuators, we focused on the feeding system of the animal. And so what we're seeing here, this is the head of the sea slug, and the feeding system is located kind of in this head region, and it's composed of a muscular hydrostat called the buccal mass. The buccal mass has a number of different muscles associated with it, which each perform different functions. And we focused on this I2 muscle, which is a broad, flat muscle kind of on the outside of this muscular structure. And why we selected this is that it's been well studied in the neurobiology literature, and we know what neurons in the buccal ganglia innervate the muscle, as well as then the buccal ganglia's connections to the cerebral ganglia. And so in this way, we can kind of think about this neuromuscular tissue circuit as an actuator, a motor controller, and a higher level controller. And with this approach, what we can do is we can actually isolate these neuromuscular tissue circuits from the buccal mass with their functional connections attached. So we start by isolating the buccal mass, and then we can carefully dissect the I2 muscle away from the rest of the musculature such that we maintain the innervation to the buccal ganglia and maintain the connections to the cerebral ganglia. And this gives us a very interesting platform to start looking at different levels of control in biohybrid robots. To begin, though, we were really initially drawn to this model animal for actuation. And so we started by looking at just this I2 muscle. So we developed a series of robots, the first of which were roughly inspired by baby sea turtles in the way they kind of scooch their way through the sand. And what we can do is we can flexibly 3D print the body of a robot using an SLA resin printer with the flexible resin. And then we can isolate the I2 muscle as an actuator from Aplysia. We can attach the I2 muscle to the robot body using cyanoacrylate. And one advantage of this I2 muscle is it's so thin that we don't have to worry about maintaining any sort of vascular system for the robot. Again, just as with the previous robots, we're able to pace the robot with an external electrical field. And you see each time we apply a stimulation to the bath, the robot contracts, and over time, it scooches forward. And I'll just speed this up so we can kind of see that scooching. And this is over the course of about a minute-long trial. So we see that we have much kind of faster performance than the first round of cell-powered organic devices, keeping in mind that this is slug muscle, and slugs are a bit slow. But when we looked and kind of characterized the locomotion of these robots over a number of different substrates, what we saw is that we were getting fairly rapid fatigue in the robot performance. So we'd get pretty quick movement initially, and then rapidly over the course of 20s of seconds, the performance would really kind of start slowing down. And this led to our next kind of engineering challenge, which was, okay, well, we have these robots with a more robust source of the actuator, right? And these actuators, I should also highlight, they were capable of functioning in both kind of the specialized support media for keeping the muscle alive, but we were also able to throw them just in an artificial ocean and have the robots perform. So much more robust than our previous robots. But they're still slow, and they still fatigue very quickly. So how do we improve the device speed and functional life? And for this, we actually took inspiration from the functional electrical stimulation community, where they've previously shown that when you stimulate the muscle belly directly in order to kind of induce locomotion, it results in fairly rapid fatigue. However, if you use a cuff electrode and stimulate the nerves that innervate the muscle, you can get improved performance and less fatigue. So we wanted to look at whether the same sort of approach would help us in our bioactuators. And can neural control improve our bioactuator performance? So what we did is we looked at the impact of different levels of stimulation on the bioactuator performance, starting with the field electrical stimulation that we've been using in all of our robots so far. And then we also looked at using a suction electrode to stimulate the nerve that innervates the muscle directly. And so we're actually able to take the nerve stump attached to the muscle, pull it up into a suction electrode very gently, and just stimulate the stump itself, which is kind of like that cuff electrode in functional electrical stimulation. In addition, with these neuromuscular tissue circuits, we're also able to look at the impact of stimulating the neurons themselves. And since this is a model animal in neuroscience, we knew that if you apply carbachol solution to the cerebral ganglia, you can actually induce fictive motor patterns that are very similar to the biting motor patterns that are observed in the animal. And so we could isolate the cerebral ganglia, apply carbachol, and induce that biting rhythmic contraction in the muscle. And as you might expect, we found that, yes, indeed, increasing the level of neural control and the level at which we stimulate the muscle improves the bioactuator performance. So using a suction electrode resulted in higher peak muscle forces than using external electrical stimulation, and using ganglia stimulation and inducing those natural motor patterns resulted in the best performance. And we also saw kind of some preliminary emerging data that this would also reduce the fatigue of the muscle. And so with this information in hand, we really came back to our bio-inspired robot roots and developed a new robot. And so this robot is roughly inspired by inchworms. And so it has two bodies, again, 3D printed using a flexible SLA resin. The I2 muscle forms a connection between the two bodies. And then we can place the cerebral ganglia in an isolation well such that we can apply chemical stimulants without applying them to the rest of the tissue. Because we are using one muscle in order to induce locomotion, we relied on frictional anisotropy to allow forward locomotion, both in the form of kind of macro scale anisotropy with these angled legs and kind of micro scale anisotropy, mimicking snake scales on the bottom of the feet. And so we fabricated these robots, and that's what you're seeing here. So we've got the I2 muscle connecting between the bodies between the two 3D printed bodies. And then up here in this isolation well is the cerebral ganglia, which we've applied carbachol to. So there's no electrical stimulation in this video. We've just induced these natural biting motor patterns, which are driving the cyclic contraction of the muscle. And this is allowing the robot to crawl forward. And what we found is, whereas the robots with electrical stimulation would crawl forward for a brief amount of time and then definitely show fatigue and eventually stop responding, with these robots, we got much better performance over longer periods of time, up until the receptors for carbachol are all kind of bound up with carbachol, and then you have to rinse the solution out in order to repeat. But after that rinse step, the robot would again respond to the chemical stimulant. So this gives us a much more robust approach to this kind of biohybrid robotic problem. But as you may have noticed, calling these robots is probably still a little bit of a stretch at this point. Right? We're missing the sense plan part of sense plan act, right? And so we really wanted to develop approaches to be able to not only kind of harvest and make use of these existing neuromuscular tissue circuits, but actually to be able to design our own motor controllers using living neurons and predict their activity towards the development of these organic robotic controllers. And our goal with this is really to be able to grow our own controllers in the dish in specific patterns in order to program the behavior of the robot. And as a new lab, as you might imagine, we got up and running on this and immediately got shut down. And so, you know, all the power and respect in the world to my PhD students, all of these experimentalists who were doing biomaterials and tissue engineering and animal work took their computers home and learned to be modelers. And took a lot of what we were learning from these tissues and the inspiration from these tissues in order to start thinking about brain-inspired computing and how we could kind of bring this as a feedback loop back to more bio-inspired robotics. Because certainly this is not the kind of robot that you can just take home and build in your kitchen. And so that's what I want to move on to now and kind of talk about our approaches in the brain-inspired computing space, and in particular, beginning with our work in developing models for real-time prediction of neural activity in the circuitry in that neuromuscular tissue circuit. And so our motivation with this work is really to be able to predict and control the activity of those ganglia in that neuromuscular tissue circuit in real time. And from a biohybrid robotics standpoint, I really think that that'll let us be able to start programming and guiding the behavior of these robots. Our solution to this was to develop a hybrid Boolean continuous model of Aplysia californica feeding. This is in collaboration with collaborators at Case Western Reserve University in neuroscience and in mathematics. We started by looking at the whole feeding system because we understand the functional outputs of feeding in the animal. And we can actually measure the neural recordings during intact behaving animal feeding. So these are the sea slugs here. You can see an adult and a juvenile. So we can get them in a wide variety of sizes, and then a little snail. And Aplysia demonstrates 3 key multifunctional behaviors during feeding that we wanted to be able to reproduce with this model of the neuromechanical system. And those are biting, which is shown here and is an attempt to grasp seaweed that is unsuccessful. Swallowing, which is a successful attempt to grasp and then ingest seaweed. And rejection, in which the animal is actually able to push out food that it deems is inedible. And in each of these behaviors, we can identify the key neurons and muscles from the behavior and the neural recording. So for example, in swallowing, the animal starts kind of by pinching the seaweed with the front region of its buccal mass so that it can't be pushed out of the mouth. It then pushes this internal grasping apparatus forward, where it then closes on the seaweed and then is pulled back and retracted into the esophagus to kind of push the seaweed back in and complete the swallow. And for each of these actions, we know the muscles involved, and we know the motor neurons and interneurons that kind of are involved in this behavior, and they can be consistently identified animal to animal. So we can see the activity of each of these units via extracellular recording, as well as the force that results in on the seaweed. And what we did is we translated each of these bursts of activity into a Boolean representation, where we're paying attention to the activity of the cell, as well as the phase and timing of these bursts of activity. Based on this, we then built a multi-layer Boolean neural network model in layers, starting from the motor neuron layer, in which we captured those key motor neurons known to be involved in feeding. We then added a buccal interneuron layer for the buccal interneurons that coordinate the activity of those motor neurons, as well as a cerebral interneuron layer, which contained neurons known to be involved in behavioral switching. The cerebral interneuron layer takes in external sensory cues, and the motor neuron layer sends motor signals to the musculature of a simplified biomechanical model, which then provides sensory feedback to the motor and buccal interneuron layers. we coupled this neural model with a simplified 1D biomechanical model that was implemented for the 3 characteristic feeding behaviors. So this is just represented here with this cartoon schematic. And the idea is that we represent the head as a point mass, the grasper as a point mass, and then the position of the head and grasper is controlled by the actions of the muscles, and springs representing the connective tissues and musculature that we're not modeling. We also simulate a seaweed that is connected to a rigid force transducer, and we can control the threshold at which this seaweed breaks during swallowing. So just to kind of walk you through what would go on in the motion of this system during behavior, for swallowing, we start at rest. The grasper is then protracted forward where it closes on the seaweed. And then the retractor muscles push the grasper back. But because the seaweed is attached to a rigid force transducer, this actually results in the head of the animal moving forward, which is the same thing that we see in the animal model in our experiments. And then the whole system kind of slips as the grasper releases and the head returns to rest. So by combining this Boolean model of the known connections within the buccal and cerebral ganglia involved in these behaviors with the simplified 1D biomechanical model, we created a model that qualitatively reproduces all 3 key multifunctional behaviors. So for example, biting is characterized by there being no mechanical stimuli in the grasper, so there's no seaweed in the mouth, but there is chemical stimuli at the lips and mechanical stimuli at the lips, indicating that there is seaweed present in the environment, and so the slug should be trying to eat. And so we see that the grasper is protracted forward and retracted back, and the grasper opens and closes, but because there's no seaweed present, no force is developed on this seaweed. And this looks very similar to the biting behavior in the animal. We can also reproduce swallowing when there's positive mechanical stimuli at the grasper and the lips, as well as chemical stimuli at the lips. And swallowing is characterized by strong retraction with the grasper closed, which results in high force being generated on the seaweed. The model also reproduces rejection, in which there is mechanical stimuli at the grasper and lips, so indicating something is in the mouth, but there's no chemical stimuli, so it's probably not seaweed. And in that case, the animal wants to reject and kind of spit out the item so that it can then go find actual seaweed. And rejection is characterized by strong protraction with the grasper closed in order to push these objects out of the mouth. Additionally, the model reproduces behavioral switching when we switch from one sensory state to another, in this case, from biting to swallowing. And so you may be sitting there thinking, well, but what does this have to do with robots, right? But by studying and modeling these known neuromechanical systems, and in particular this system, which is the ganglia and muscles that we're interested in using in our robots, we now have a basic model that we can take back to our biohybrid robot design in order to model the activity in the actual neural circuitry that is part of our neuromuscular tissue circuit. And possibly even then kind of hijack these sensory nerves to provide these different sensory cues and induce different behaviors in our robot. And that's work that we're coming back to now that our lab is reopened and up and running. And so in addition to developing these neuromechanical models for neuroscience, we can also take what we learn about how these tissues grow and interact and use that as a source of inspiration for more bio-inspired approaches in computing. And this is work of my PhD student, Kevin Dye, who was very interested in looking at whether the sparsity and the patterns of connections in biological neural networks could improve performance in artificial neural networks. So in traditional artificial neural networks, many networks are implemented where they are fully connected, right? So we have a layer of artificial neurons, and each neuron in the input layer is connected to every single neuron in the hidden layer and down through the layers, with all of the neurons connected to all other possible neurons. And of course, this isn't how biological brains are organized, right? Even though we have kind of millions of neurons and billions of connections, the relative connectivity of biological neural networks is still relatively sparse, right? Where each neuron is only connected to some of the neurons in the next layer. And so what we were looking at was whether we could simulate growing these neural networks in order to identify connections that could then be used in artificial neural networks. So the approach that Kevin developed was to start by initializing a spatial growth map with the neurons. So here we have the input layer neurons of his artificial neural network in blue, the hidden layer neurons in red. And then he specifies for each neuron a number of axons and growth parameters, a growth angle and a growth rate, selected from a stochastic distribution. He then simulates the growth of these axons until they reach the next layer of neurons. And then once they're within a certain radius of another neuron spatially, they form connections. And then those connections are summed to develop the weights between different neurons. For testing this approach, we tested this on the MNIST dataset, which is a benchmarking dataset in artificial neural networks, looking at image classification. So the goal is to be able to predict the correct digit for a handwritten picture of a number. And so what does this look like in practice? So first we start with the spatial growth map, and we grow the axons over a period of steps. And so here you can see the tip locations of those axons growing out during growth until they start consolidating around the hidden layer and they've formed those synapses. We looked at 4 distributions for selecting growth parameters: a Gaussian distribution centered at 0, a Gaussian distribution centered at 1, a uniform distribution between 0 and 1, and then Kevin also developed this reflective method where if a neuron tries to grow outside the bounding box of the tissue, it's reflected and it grows back in, so that we didn't lose these connections out into space. For each of these methods, as you might expect, the axons with smaller growth angles connect in fewer steps. And then over time, the axons with these higher growth angles form their connections as we increase the overall connectivity of the network. But we're looking at relatively low connectivities percentages, so pretty sparse networks. Once the axons have all grown, the network weights are set based on the number of connections between any given pair of neurons. So it is possible, because we're using these stochastic distributions, that an axon may form more than one connection with, or a neuron may form more than one connection with another neuron, in which case the weights of those are randomly selected and then summed. And then all the unconnected weights are set to 0. This connectivity map was then imported into a PyTorch framework using a modified linear layer module, and a mask was applied in order to keep the unconnected weights set to 0 during backpropagation. The neural networks were then trained on this MNIST benchmarking dataset, and we do see that kind of this sparsity is maintained during training, as well as the fact that the training does change the weights within our network. So here you're seeing the untrained weights for each of these models that Kevin looked at. And then following training, we see that we've updated the weights in each of these networks. And this interesting banding effect on this diagonal, we believe is just an effect of this physical spatial map, because those axons with low growth angles connected first. And what's really interesting is that for each of these distributions, what Kevin found is that the GRINS networks were able to achieve higher test accuracies compared to that fully connected control network at any of our connectivity percentages kind of higher than 20% connectivity. So with relatively sparse networks, we're able to achieve higher test accuracies. And now Kevin's looking at kind of updating this growth model to actually capture the chemodynamics involved in axon growth, as well as looking at now using it for robot control rather than image classification. And so this is a kind of a picture of the work that we've been doing so far. And I just want to wrap up by talking about some of the future directions in biohybrid and bioinspired robotics that we're looking at. So building on our work in developing these renewable biocompatible structures, we're now very interested in how we can integrate them more effectively into our bioactuators, beginning with developing fabrication techniques to really be able to combine these tunable threads with hydrogel and protein-based materials. In addition, we have ongoing efforts in creating living adaptable robotic actuators, both in extending our neuromuscular tissue circuits to include sensory organs so that we can have a little bit more of that sense, plan, act paradigm in our robotic platforms, as well as improving our techniques in the tissue engineering approach to developing these bioactuators to really be able to grow the architectures that we want and develop more complex robotic structures. And now that we're back in the lab, we are working on our development of these living organic controllers. And my PhD students, Ashley and Kevin, are collaborating to really quantify actual growth distributions from these neurons in culture, both to apply those to the artificial neural network approaches, but also to be able to then plan how we're going to fabricate neural circuits with the connectivity and capabilities that we want. And this is really all towards our long-term goal of creating these renewable, biodegradable, autonomous robots. And with that, I'd like to again thank all of the students and collaborators who have been involved in this research. There's a lot of hands that are involved in these projects along the way, as well as our funding sources. This work is funded by the NSF as well as the NIH. And I'd be happy to take any questions. Thank you.